We left off in part one of this lecture talking about pastries, and I'll spend a couple more moments on that just to remind you the wonderful thing about pastries is the laminar or flaky texture of many pastries, and that arises from the coating of the dry particles of the batter with some fat, right, or some oily substance that prevents them from merging. And then as you flatten and stretch, fold and refold and stretch that dough, you get this very wonderful laminar structure that's alternating layers of batter and butter or oil or shortening. Reminding you again that the water content here is something that should be controlled very carefully. That is to minimize, not eliminate, but minimize the water contribution, and that just keeps the dryness and flaky texture. And then, of course, when we heat that up, we see extremes here, right, of more of a cake type structure here, more of perhaps a croissant type structure here with these sort of, or pie crust, sort of flat but not too thin plates. And then here, when we've really folded and stretched that dough out, we see something more like phyllo dough here, which is this very wonderful texture that occurs as the different lamellae or flakes or layers, if you will, puff up and separate from one another. And they're again allowed to do that because they've been coated with that fat. Just as examples, here's an example of baklava. There are many local varieties of this all throughout the Mediterranean and the Middle East, and just layers and layers of this very finely laminated dough. And this is all from one piece of dough that's just been folded and rolled and folded and rolled many, many times. And then, of course, being soaked in honey doesn't hurt either. So phyllo deriving from the Greek for leaves. Croissants, you have something similar. Nothing quite so fine, but you can see that this in this croissant dough we have folded it into lamellae or layers, not nearly as many as you see in phyllo dough, but still this gives rise when we bake that to this wonderful wonderful layered texture of the croissant. Okay, so again, we've talked about how much of baking depends on the fermentation carried out by the yeast on the sugars of the ground flour, but fermentation takes a lot of other forms, and I want to begin discussing alcohols today. We've already discussed lactic bacteria. Those are organisms that help us convert um, lactose or other sugars into lactic acid, providing a wonderful acid flavor, a sort of a sour flavor, which we're familiar with from yogurt, certainly in yogurt, some cheeses as well. I'll just mention here too, Leuconostoc. This was a bacterium that we encountered that is used in many European sour creams and creme fraiche, for example. But beyond the lactic fermenters, let's think about the alcoholic fermenters. And of course, we're probably familiar already with bakers or brewers yeast, Saccharomyces cerevisiae. This is the yeast that we use in baking, and it's also close relatives of what we use in baking are used in the fermentation of grape juice to wine. There are other alcoholic fermenters, Zygosaccharomyces, used in the preservation of many foods, and it's a wonderful yeast because it has a high tolerance for various components that we'll see in sugar. In our cooked products, as well as for ethanol. So high acid content, high sugar content that we might see in our food products, this organism can survive. Another important idea to mention here in the alcoholic fermenters is that those are tolerant to ethanol, and various subspecies of Saccharomyces and Zygosaccharomyces are tolerant to relatively high or low concentrations of ethanol, and this is going to determine how far one's fermentation goes, or this is one component of how far the fermentation goes. Some species of yeast can tolerate ethanol concentrations of up to 20%, whereas you're probably familiar with the common percentage in beer is something between 3 and 5%, wine maybe up to 10 or 12% ethanol. That, among other factors, is controlled by the variety of yeast or alcoholic fermenting anaerobe that one is using. There are lots of other organisms that we encounter in our fermentations. Some of them are directly fermenting, some of them are carrying out other functions. These are molds that I've listed here. Of course, the penicilliums we're familiar with from the cheese discussions we've had, and we'll talk about these others as we move along through the course. Aspergillus, we'll see that that's used in the fermentation of soy beans to soy sauce. Rhizopus, we'll talk about tempeh, also used in the fermentation of some alcoholic beverages. Again, soy cheeses, so more interesting soy products, mucor and actinomycor. These are wonderful molds. We'll come back to this one involved in sweet wines at the end of today's lecture, Botrytis. And then there are other organisms that pop up in fermentations that are not so useful. Clostridium, for example, an organism that produces botulism toxin, tolerates low acid but is also an anaerobe. So in the conditions where we'll see that where we're preserving food, and we've already talked about anaerobic conditions, this organism thrives if the acid concentration is low, that is to say the pH is relatively high. So we have to be careful, for example, when we are preserving foods to maintain proper acid conditions. And we can do that in part by controlling what other organisms are present. So let's say a little more about fermentation as a preservative, and mostly here just to introduce some of the food products that we're maybe accustomed to that are actually fermented foods, that is to say they've been partially degraded with the help of these fermenting anaerobes. So these are some of the things that the anaerobes control. pH and ethanol, for sure. Also, we can control the growth of certain microbes with the addition of salt. So these fermenting organisms break down the food, as we've seen, but they also, as I mentioned, prevent other organisms from establishing themselves and taking over control of the fermentation or the food. So some examples: gravlax. This is a Scandinavian dish, the name meaning something like grave salmon or dead salmon, so preserved salmon. Olives need to be fermented in order to be palatable. Sauerkraut, this is again Northern European preserved cabbages. Pickles, we're familiar with cucumbers that have been partially fermented, although most of what we eat in the supermarket nowadays are created through a different process, industrial processes. Manioc, an organism, a plant grown in the tropics and subtropics of the Americas, also known as cassava. The pulp from this vegetable fermented to be made more palatable and incorporated in many foods. Poi, close to my heart as I'm about to head off to Hawaii next week, which is the product of the taro root. Poi can be fermented or not, but again, I think one will discover that when we cook these foods, if we, especially before the advent of refrigeration, if we don't finish them right away, they will ferment. And so some of the wonderful food products that we enjoy today derive from the accident of fermentation by endogenous anaerobes present in those foods. Kimchi, of course, this is again cabbage, although there are wide varieties of kimchi types and recipes, but essentially fermented cabbage. And this lower picture here is kimchi. We don't have time to do justice to all of the varieties of kimchi, but these are some jars of fermenting kimchi that you can see, just so you can get a sense of how this might be performed in some circumstances. Although, of course, there's lots of industrialization of all of these, preparation of all of these products now. But they all started out, at least, as foods that were preserved via the fermentation by anaerobes that produced changes in the conditions of the food that prevented other organisms from taking place and spoiling, quote unquote, spoiling the food. Back to olives. I told you that this product had to be fermented in order to be made palatable. Well, part of what happens is I'm sort of getting at that in this image here. It has to do with the degradation of phenols or polyphenols, which I think I might have mentioned before, and I'll come back to again later. Plants often produce molecules called phenols that are very unpalatable, and this is frequently a defense mechanism to prevent other animals from consuming that product and maybe interfering with the reproductive cycle of the plant or what have you. In this case, oleuropein is the bitter phenol that is broken down by fermentation by various other organisms. The phenols have a variety of purposes, as I mentioned, maybe in some cases defenses, other cases defending against oxidation. And we'll talk more about antioxidants in the future, but these are basically, you could consider them as dumps for reactive oxygen species that could cause damage to the proteins or the DNA of the organism. Instead, compounds like this one absorb that oxygen, and that's essentially what an antioxidant is doing in most cases. So let's remind ourselves about alcoholic fermentation. I've told you how when we break down glucose to pyruvate, this generates a little energy. However, in the presence of oxygen, we can perform something called mitochondrial electron transport that generates a lot more energy. But in the absence of oxygen, under anaerobic conditions, we stall, right? And so the energy is transmitted to the mitochondrial electron transport chain by NADH, but in the absence of oxygen, we can't use that NADH. What the upshot is that the NAD plus required for conversion of glucose to pyruvate during glycolysis isn't present, and so glycolysis also stalls. So not only are we not getting energy out of mitochondrial electron transport, we're also not getting energy out of glycolysis. And so fermentation, again, is the process by which we kickstart glycolysis. Conversion of pyruvate first to acetaldehyde and carbon dioxide. This is important for our beer fizziness and our champagne fizziness. Production of CO2 by pyruvate decarboxylase, and then conversion to ethanol, acetaldehyde to ethanol, by alcohol dehydrogenase. Ethanol, of course, is alcohol. Now, the key here is that in this reaction, alcohol dehydrogenase reoxidizes NADH to NAD+, which of course now is ready to go and turn over another cycle of glycolysis. So that's how we keep energy production going, and that's how the yeast can survive under anaerobic conditions. Now, I should be careful to say that yeast can carry out alcoholic fermentation under aerobic and anaerobic conditions, so it's called facultative anaerobe. Okay, a little history. So we're going to spend the next couple of minutes in part 2 of this lecture discussing wine, and we're going to continue with wine and into beer and hard liquor next week. The earliest evidence for the fermentation of grape juice to wine is about 7,000 years ago. And I love showing you these little history slides. This is from a paper published in Nature, oh boy, starting to be quite a while ago, showing archaeological evidence for the presence of tartaric acid. So, of course, tartaric acid we also know as what? Cream of tartar, right? An important leavening acid, as we've already discussed, the active ingredient in baking powder, for example. And of course, cream of tartar is something that we use in various baking recipes. But tartaric acid is found in high concentration in grapes, and the wonderful thing about having this high concentration of acid in our grape juice is that it stabilizes that product against bacterial contamination. So that's an important feature that's important for the preservation of wines for a long period of time. Now, historically, wine casks, wooden casks, were not used for storage of wine, but instead stone amphora. You'll hear this word a lot when you, if you watch your, what do you call them, TV shows about sunken treasure, right? You'll see this a lot in the stone amphora that are left behind in the rotted vessels at the bottom of the Mediterranean. In any case, very useful for long-term storage of wine. Now, of course, the wine casks or wooden casks were introduced maybe 1500 or something less than 2,000 years ago. And while wonderful and easily made and allowing the ramping up of wine production, perhaps simplification of wine production, they're just not as good wooden casks at preserving the wine as those stone amphora were. So more is required, I guess, for the preservation of that wine than the low pH that Tartaric offers. And so what we saw in wine was a decline in quality. This is the assertion of the author of your text, that lasted more than 1,700 years before the introduction of, as we'll see, sulfur dioxide or sulfur as a preservative. In any case, let's turn to the evidence for this 7,000-year-old wine cask. Here's again the structure of tartaric acid. All I'll point out here is that here's an acid group, a carboxylic acid. Tartaric acid happens to have 2 acid groups, so it can donate 2 protons overall. This is a cutaway of the amphora or vessel that they discovered. And these are, over here, I'll take a moment to talk about this. This will look familiar to the organic chemists in the audience, but to others of us probably not so much. This is an infrared spectrum of extracts on the top here from the wine amphora from Haji Firuz. Let me back up and say that the archaeological site that this amphora was discovered in was Haji Firuz, which is a mountainous region in northern Iran. A Neolithic, right? We're talking about the Neolithic era at this period. And there's also another site here, Malkata, that they have a similar amphora similar product extracted from wine amphora. And then here's calcium tartrate. So this is the pure chemical that they're using as a reference, right? So in this experiment, what we're doing is looking at infrared spectra of 3 different samples. One's a standard calcium tartrate pulled off the shelf in the lab. One is a known wine sample from another site, and the third is the stuff scraped out of the amphora at Haji Firuz. And what they saw for calcium tartrate is that there's this very big peak here at about 1,600 wave numbers. I'll talk about this again in a minute. And that matches, that's a distinctive feature of the calcium tartrate sample, matches the spectrum from Malkata, and then our sample from Haji Firuz has the same peak. Now it's a less obvious component of the spectrum than in the other samples, but very clearly there. So that's their main supporting evidence for the fact that this was a wine cask, and this places the invention of fermentation of grape juice to wine at about 7,000 years ago. Lots of great stuff happening about 7,000 years ago, right? Invention of farming and selection of barleys and wheats to make wonderful 2-row and 6-row wheats that we use for brewing and baking, and concomitant domestication of cattle and introduction of the milks, and also fermentation. IR spectra. So let me back up and say that a spectrum is any kind of measurement wherein we're looking at the interaction of light with matter. We're all familiar with spectra. Our eyes are some of the best spectrographs around, or spectrometers around. You hold up a bottle of Kool-Aid and you see that it's red or purple or blue, and you see that if you stir in more Kool-Aid, it gets darker red or darker purple or darker blue, right? That's a spectrum, right? That tells you that the light that's passing through that sample, for the sunlight that's passing through your Kool-Aid jar, all of it is being absorbed except the blue light, which is passing through. Well, here we're not looking at visible light, we're looking at infrared radiation, and what we see is that at certain wavelengths, or colors, if you will, of the infrared spectrum, some light is absorbed more effectively than others. And without getting too much deeper into that process, in infrared spectrum, what we're talking about is the vibration of chemical bonds. And it just so happens that there's a characteristic vibration that corresponds to the absorbance of radiation at 1600 wave numbers that we see in calcium tartrate or samples that contain calcium tartrate. So again, I'm belaboring this point, but it's really cool. We were able to determine that this jar, we scrape the residue out of the jar, throw it in our infrared spectrometer, and we see that indeed there's calcium tartrate. Now, you don't find high concentrations of calcium tartrate elsewhere besides in grapes. So, ergo, wine amphora.